Feeling nervous? Oh, geez, I had to repeat that for the recording. But I tell you what, I look forward to this lecture every year just so that I can say that as my intro. If you're feeling nervous. Oh, yeah. Okay. Let's see if there's any questions going on in the chat group. No, you guys are certainly quiet this morning. Is it you're just feeling overwhelmed by all of the like you're just astonished by the actual lecture content that you're learning, and then that's it. Is that right? Yeah, I thought so. I can tell, you guys have been waiting for this lecture your whole life. And look, this lecture will change your life. Like, guaranteed, it will change your life, right? You will go from, if you've never really felt that you had sort of, a purpose in life or anything like that, then I'll tell you what, after this lecture, you'll go, my purpose in life was to listen to this lecture. Okay, let's get on to it. We've got a fair bit to get through. I'm sure you all have purposes in life other than this lecture anyway, but can't resist saying that as well. This is the learning outcomes. Remember, Reed, Martini, let me see. So, yes, so Teo, I already mentioned that. That's all been updated. I think you may have arrived after I'd mentioned that. Yeah, I was able to get the tech people to do it. So basically what happens with this unit is I go into the lecture recording area every Monday morning. I record all my lectures, and then I get assistance from the tech team to put them up. So apologies about that. I did notice that 5.1 was uploaded twice, but we have to put the request through to the tech people, and in very typical bureaucratic fashion, they take a little bit of time to sort of get to those jobs as well, which is really annoying. But no, glad it's fixed. It's all up there. So just a couple of housekeeping issues. So the lab one's done, everyone. Unless you've got special consideration, I mean, even then it's really hard because we can't just redo the labs, right? So lab one's done, and if you missed out on anything, then unfortunately you've missed out on 5% if you've missed out on a lab, and that's because the we're not running lab one anymore. We're under lab two now. Okay. So everyone, I know some of you have had some things going on in life and all that stuff. Look, it really is, university is a lot more like work than it is high school. So if you don't turn up, you just don't turn up and you miss out. Unless you've got really valid reasons for special consideration that you've got to apply for formally throughout the university, which I don't recommend doing all the time because you get a record of it, so we can actually see when you've got a big important assessment down the track, and if you've repeatedly applied for special consideration, everyone can get a medical certificate nowadays. It's pretty easy, right? You can get it online. It's a joke, right? So what I'm saying is, for those of you who just missed out on your lecture, on your lab, or you had something else socially that popped up, or so I know a few of you have messaged me, you just said, oh, for certain reasons I couldn't attend, then I'm really sorry. There's nothing I can do, because unless I was able to schedule you into a blank spot, which there's hardly any in these lab groups with basically 600 students in the whole unit, with all the lab classes being really full, I really need everyone to take more responsibility for their own timetable, because I'm getting like about a dozen emails every day about timetabling issues and trying to get people into groups and stuff. So please take a bit more accountability for your own actions, okay? As soon as I said that, a whole bunch of people left. You've just got to submit it as a PDF file, Khan. So as long as it's a PDF file, that's fine. Okay, these are your learning outcomes for the day. So you've got to describe that. You're welcome. And I hope I wasn't too harsh by saying that, but does everyone get what I mean? Like, I can't really, there's a lot of related stuff that I can't do, and I just don't have access to your timetables or anything like that. And if you miss your lab, then and there's no other spot that I can try and put you in, then we can't just create a new lab for you. Okay, so okay, great. So that's the hard talk out of the way. Let's get into it. So this part of the lecture, you got to describe the anatomical and functional divisions of the nervous system, sketch and label a typical neuron. I'll tell you what you need in regards to what stuff you should be focusing on. Classify neurons according to their structure and function. Describe the locations and functions of neuroglia or neuroglia in the nervous system. Describe the events in generation and propagation of action potential and the factors involved in propagation, speed of action potentials. You only need to know the basics of that, okay? There's a lot going on there. I don't want you to get too worried about that because it can get tricky. You need to be able to briefly identify the mechanisms involved in synaptic activity, super easy stuff at that point, and explain how resting membrane potential is created and maintained. Okay, so as you guys know, I'm going to skip through a lot of this stuff because we go through it more in the recordings. I'm going to cover most of the really important stuff, okay? And you stop me at any point if you need me to focus on a couple of things. Otherwise, I'm going to skip past slides. So if you need me to go back and explain something a bit more, then you just say, hey, Greg, can you go back? Okay, we all good with that? Yep, fantastico. We're all a bunch of funsies. Okay, this is good. So just remember, there's two different types of nervous system. You got your central nervous system and your peripheral nervous system, right? One's your brain, your spinal cord. The other's everything else. So, and what's really important. This is definitely like going to be a multiple choice question or something, or a true or false one on the exam. You should know the difference between neuroglia and neurons. Neuroglia are not neurons themselves. They're glial cells, and what they do is they support neuronal function, but they're not neurons themselves. Okay, like I said, you got the CNS and the PNS. You got to remember these definitions, okay? They're really easy, right? Afferent is input coming in, so that's a sense that you pick up. Efferent is output. So when we talk about efferent, we're talking about somatic or autonomic. Autonomic is obviously ones that are just going to work constantly, and then somatic are ones where there's going to be a bit more choice in regards to the way that you might move your limbs and things like that. So an enteric is basically your input and output from the gut. Your gut, as we'll talk about later on in the semester, is a hub of activity. It's basically the biggest part of your nervous system, of your immune system as well. There's so much research that's gone into understanding the link between the way we function, the way we perform, and the way our microbiome of the gut bacteria and all that we have that contributes to our overall function as a human, which is really, really cool. Okay, so like I said, somatic nervous system controls all your skeletal muscle contractions, your limbs and things like that. That's both voluntary and involuntary. When we talk about involuntary, we're not talking about the beating of your heart, we're talking about reflexes. The autonomic nervous system controls a lot of your heart function, your glandular secretions, all the stuff that's constantly going on in the background that you don't have to think about. So it's autonomic because you don't have to tell your heart to keep beating. Imagine how lame that would be if you just had to if you forgot how to breathe or something. So you don't have to keep saying to yourself, all right, breathe in, breathe out, breathe in, breathe out. You don't have to say that because that's all governed by your ANS, not your SNS. Okay, the enteric nervous system, as I said, it is related from the information you get from your gut. This is a really good slide to sort of to like sketch out, and just maybe print it out, frame it, put it up on your wall, go to IKEA. Actually, IKEA, how they have those really lame sort of pictures that they're just things they've ripped out of a magazine when they're doing those room displays. Does everyone know what I'm talking about? Yeah, and then they'll have like things posted on a fake laptop or something. What you can do, if you're really cool, what you'll do is you'll print this out and you go and put this in, on laptops in IKEA and stuff like that, those fake laptops. So it just looks like everyone's studying Bio 10,004, right? Okay, this is a really good one because what it does is it breaks up everything, right? This is a pretty good one for you to remember. Okay, so in your neurons, obviously there's things that you've got to remember. I'm going to tell you exactly now what you need to be able to remember. Now, if I show you an image here, you should be able to identify the dendrites. You should be able to identify the cell body, and you should know that's a nucleus. You should be able to also know that that's the Golgi, for example. That you've got, you should be able to identify your basic cellular organelles. But having said that, that goes for any cell, right? What you should be able to understand is that's the cell body. Over here's your dendrites, but you're going to, get that information. Here's the axon, and there's the axon terminals, and really it would be nice if you could remember telodendria, which are the things that are going to transmit that, come off the actual axon and be able to pass that through the axon terminals, where it's innervating and yet another cell body over here by interacting with the dendrites on that cell body. That's all you need to know. There's so much going on here. I'm not going to ask you, expect you to know what the axon hillock is, for example, or identify what the axolemma is or anything like that. You'll do that when you do more advanced neuronal units in the future, if you do them. For this, I just need you to at least be able to identify those three major components, and for extra points, the fourth one that I mentioned as well would be ideal. Okay, so here we've got, as I mentioned, you've got, you should be able to remember the basic structure. I'm going to skip through these. You should be able to remember the basic structure of these neurons, right? You should be able to look at something and say, that's an axonic neuron, that's a bipolar neuron, a unipolar neuron, and a multipolar neuron. You should be able, and what you'll notice is these are all just different, I'll put my cursor on. These are all the same thing, really, when you get down to it in regards to this. In regards to, like, they've all got their cell body, their dendrites, their axon, the axon terminals. They've just got it in different locations, really, haven't they? So what you should be able to know is have a look at that. And then also you should be able to go back and be able to really have a good idea about where those particular neurons are associated with, or what they're actually associated with as well. Like multipolar neurons, for example, basically all motor neurons that control skeletal muscles are like that. A lot of sensory neurons are unipolar. Axonic neurons are found in the brain and special sense organs, and your bipolar neurons are found in special sense organs. So again, that's all you need to know for that. You should be able to identify them by sight, and you should be able to say roughly what they're doing. Okay, a couple of basics here in regards to sensory neurons, motor neurons, and interneurons. So you should be able to remember the basic functions of these as well. Interneurons are pretty cool because they're the ones that a lot of people have a lot of interest in when they start learning about the brain, because they're associated with memory and things like that. Neuroglia. You should be able to know this. We've already talked about this many times. We talked about it when we were talking about organization of the body at the tissue level. Neuroglia essentially are the cell types that will look after the nervous system, but they're not nerves themselves. So in the peripheral nervous system, that's satellite cells and Schwann cells. In the central nervous system, it's mostly astrolites, epidermal cells, oligodendrocytes, and microglia. Pretty cool stuff. I'm not going to go through this in great depth, and I'm not going to expect you to go give full description, say, of the development of astrocytes or anything like that in the exam, but I would like you to be able to know what is what's what in regards to the particular types of cells that make up neuroglia. All right, this one here is a big one, but it's not that big in regards to the fact that I'm not going to ask. You might be asked to explain the basics of this, so just know the basics of it, but I'm not going to ask you really major things here in regards to membrane potential. Remember, your nervous system functions electrically, right? And that does that by passing on ion-based movements. The way you change charge of a neuron, we're basically passing electricity through neurons, right? The way we do this is the same way that batteries work in this regards, right? Batteries obviously not as sophisticated as this, but it's a very similar concept in this case here. You have polarization of a membrane. So you have charge because you have one end that's positive, one end that's negative, just the same as what you have in a battery, right? So the way that we actually change that charge and we charge it up so that it can be able to generate this, move this electricity across, is we basically want to shuttle the actual ions around here. And that's where the plasma and sodium pump comes from. You should know that the resting membrane potential is minus 70 millivolts, and that the way that we change that around is by using ATP, so it takes energy. What we do is we use that sodium-potassium exchange pump that we talked about a few weeks ago in terms of membrane dynamics, right? We basically eject 3 sodiums for every 2 potassiums that's brought in, and what that does is that changes membrane potential, right? We'll just move across here. So there's a couple of things here that you need to know the difference between. Graded potentials are not the same. Action potentials are just one big burst, right? So it doesn't matter what the stimulus is. An action potential is constant. A graded potential is something that, as the name suggests, it's graded, so it can actually vary in regards to the amount of excitation. But this is what I mean here. So you've got your membranes are essentially polarized at this point. You've got a negative charge in the cytosol, positive charge in the extracellular fluid. Over here, what we do is we get the sodium potassium pump going, and this way here we start to actually get depolarization of that membrane there as well. As you can see here, this drops the charge, and so on, and this happens across the membrane. You can see it's actually a movement, a gradient that we have going over there as well, as you get a spread of the sodium ions moving across. What this does is this transmits the actual stimulus and allows that sort of that impulse to be passed through the neuron. But what happens here is you go through a process where the chemical stimulus is applied, and the chemical stimulus that we're talking about here is this particular part here where the gate gets opened up. Now this is going to take ATP, and then what ends up happening is you have depolarization of the membrane, so sodium starts coming in, starts to depolarize it. Once it hits the maximum potential that it actually needs, what will happen is basically the chemical stimulus gets removed, the gate gets shut, and then the repolarization occurs. In this particular case here, then you've got, if you need hyperpolarization, then again, the same thing happens, but it's the opposite, right? There's a couple of differences between, like I said before, graded potentials and action potentials are different because a graded potential is basically a more of a gradual sort of effect, whereas action potentials are all in. That's all you need to remember for graded potentials, right? So yeah, I'm not going to ask you to have a look at this in the exam or anything like that. You definitely will be asked in the quiz a little bit about differences between gated and action potentials, but that's only really if you're lucky enough to get those questions. Remember, our quizzes are actually comprised of like a massive question bank, which I write myself, and then a lot of it is really just they're all based on the lectures, and then you get 10 randomized questions. Oh, by the way, as anyone in this lecture, were they impacted by the Canvas sort of update that happened the other day and screwed around with quiz 4? Was anyone impacted by that who hasn't contacted me already? Because those of you who already done it before, I think it was around, there was an update to Canvas that happened around 3 o'clock on Monday. And what it did is it unlocked a whole bunch more questions. And I think what it did was it sort of merged it with last year's quiz because they were both named Quiz 4. So what I did was I was able to go back and fix it once I noticed it happened. But is anybody else impacted by that? Yes, I've already replied to your email, Taya. So yeah, so if anyone had 18 questions instead of 10 and you haven't, then send me an email. And if you like, I can actually correct it for you. I can reset it for you and you can have it. So just send me an email. Yeah. I think I've already replied to you, Rashwinder. Have I? It's up to you. It's up to you. So if you got 18 questions and you're happy with the 18 questions that you did, then you can keep it if you want, because that's out of That's basically 3%. So it doesn't matter if it's 3% of something that's 18 questions is still going to be graded the same. So if you think you did pretty well in the 18 and you're happy with that, if not, if you want to limit it down to 10 and it'll be 10 extra randomized questions, then by all means, you can send me an email. I'm really sorry about that, everyone. It's something that it hasn't happened since I've been at Swinburne, but that only seemed to impact that one quiz as well. So it didn't impact anything else. All right. So like I said, action potentials are continuous propagation, and basically you should know that they mostly occur in unmyelated axons. The reason why they happen in unmyelated axons is unmyelated axons don't have that protective sheath, right? So that means that, remember, when you myelate your axons, it means they've got greater insulation, and it's almost like, if you want to think of the what's the internet we're running on at the moment? It's not cable. It's the whatever it's called, broadband, right? So, we're running ours in Australia through the copper network, and it's really slow and crap. So it's almost like an unmyelated axon is like running your broadband, your NBN through your copper network. So it literally just is nowhere near as fast. When things are myelated, they can go at warp speed. When things are unmyelated, they're slower. And this is where you have diseases. There's a number of these where there's the demyelization of the actual these autoimmune disorders, which will eat away at the myelin sheath of nerves. CIDP is one. Chronic inflammatory demyelating polyneuropathy is one that comes to mind. And what that does to people is they start to move almost like they've got MS or motor neuron disease. And that's because there's a decreased speed of the actual throughout the actual nerve. So what happens is those particular nerves need this constant propagation, right? So unmyelated neurons need constant propagation, and that's because, and usually they'll be involved in certain tasks like skeletal function and all that sort of stuff there as well. So they're always getting this impulse. Okay, the way that action potentials are a little bit different is, as we said, you just get this big burst of steam that comes in, starts to depolarize it's an immediate depolarization of the actual of the membrane, and then you missed the first 10 minutes. Yeah, so Amelia, that's been corrected. That's all up on there. So I think I noticed that when we put it up, and I had to get the tech team to put it on, so they put the wrong link on. But yeah, if anyone wants to check that and let me know if there's any issues, and I'll get them to fix it again. But that should be corrected by now. Okay. So I'm going to skip past this. I'm just going to skip past these because I don't want you to get too bogged down with that. You should know these couple of terms that you should know. Very basic stuff. Presynaptic neuron sends the message. Postsynaptic neuron receives the message. You're playing dominoes here, right? Presynaptic neuron is the domino you hit. Postsynaptic neuron is the one that gets hit by the one that you tapped. We have two types of synapses. There's electrical and chemical. Electrical is what we were just talking about before, whereas chemical is like the neuromuscular junction that we were talking about last week. And that's where you use neurotransmitters. Yep, no, that's good. No, Amelia, my apologies for the fact that it happened. It was a bit of a uploading all the new material and stuff last Monday on Monday was obviously not ideal because we had a problem going on with the whole Canvas update and stuff. So, but everything should be smooth sailing for the rest of the semester. Okay, these are the neurotransmitters. You don't have to remember every one of these, but you should. But I definitely want you to at least know acetylcholine because that's the most common one. Quiz 4 grades have not been published yet, Malika, and that's because there are still people who had special consideration who are doing it or who have learning plans which give them extra time. So at the moment there's still students actually doing any of that stuff, we will not let release any marks because we don't want to open the doors to collusion. Okay, let's get into the second part with spinal cord and brains. This is the part that you did not get the recording for, but it's up on there anyway. It's on there now. And that's an absolute banger, this one. Like this one, if there's, like I said, when I was writing this, I thought to myself, if I'm ever sure about anything in my life, I'm sure of this lecture, okay? And that's because there's so many cool things. Discuss the structure and functions of the spinal cord and describe the three meningeal layers that surround the central nervous system. Also makes sense why that everyone's heard of the disease meningitis. Has anyone heard of that one before? And everyone goes, ooh, when they hear it. It sounds bad. Yeah, that's because it actually can really stuff up with your spine or your brain as well. So it's infection of your meningeal layer. Your meningeal layers surround your central nervous system and protect them. You should be able to explain and identify the structures of the spinal cord and you should know what the difference between white and grey matter is. You should be able to describe the distribution of spinal nerves, and how that in the different parts of the body that they nerviate. You should be able to describe the reflex arc, really easy stuff. Name the four regions of the brain and brainstem and are able to identify the locations and function of each. The parts of the brain are what you're going to have to look at Martini on. We didn't add them in on here because we covered the part of the brainstem. I'm not going to ask you tricky questions about the brain anyway. I will expect, though, that you should be able to identify things associated with the brainstem. But I'm not going to show you a picture of a cadaver's brain and say, identify with all the lobes and all this sort of stuff as well. That's just too nasty. You should be able to identify the major anatomical subdivisions, including the cerebrum, and then you should be able to understand the difference between the main gyri and sulci, because they're just like literally the grooves in the landscape of the brain. Okay, your spinal cord is protected. It's protected by your spine. I had a bad fall on the weekend and absolutely stuffed my sacrum up. So I can definitely tell you that it's no joke. Your stuff. Anyone here has had a good fall and injured any part of their spine, you can tell that immediately everything clams up, and that's for good reason because your spinal cord needs protection. Spinal cord is only around 45 centimeters long in your average adult, and actually it stops lengthening at around age 4. It will stretch out a little bit, but it stops lengthening around age 4, even though your vertebral column will continue to grow. Fantastic, huh? Who ever thought about that? You can go up if you've got a 4-year-old or if you've got a 5-year-old sibling or nephew or something like that, just you can go up and say to them, your spine doesn't grow anymore. Your spinal cord stopped growing. Do you know that? And they'll look at you and say, what's a spinal cord? Okay, you have 4 regions, as you know. We covered all this already when we were talking about the skeletal system before. You already know this stuff. I'm not gonna quiz you guys too much on this because I've already covered this stuff when we were talking about the skeletal structure. Okay, so with the spinal meninges, we've got 3 different membranes that surround the spinal cord, and these are continuous up until the brain as well. So the brain has its own arachnoid matter and pia matter. Now, the actual naming of this, I'm pretty sure this is Latin. Dura means hard, arachnoid means spider, and pia means mother. So you have your hard layer, your spider layer, and then you have your mother layer. Again, like I said, there's some terms in anatomy and physiology that just people were just really describing what they actually saw and what they thought things looked like when they were doing early dissections back in the bad old days. Some of these names are a bit silly, but you do have to remember them. Just remember from the outer all the way to the inner, it's dura, arachnoid, and pia. Functions of your spinal cord, as we mentioned, you guys should know that protecting the spine and nourishing it with nutrients. Here we go. Here's your meninges, right? Here we go. Here's the dura matter, the arachnoid matter, and the pia matter over here. So the mother protects the spine, the spider wraps around, and then the hard layer is on top. Oh yeah, there you go. Okay, and so when I say know the difference between grey and white matter, this is really it. I'm not going to quiz you guys a lot in the exam on this. You should know that grey matter is your cell bodies of neurons, your neuroglia, and your unmyelinated axons. And your white matter can just comprise mostly of axons. They're either myelated or unmyelated. It depends on the function, right? Okay. When we talk a little bit about grey matter, we talk about its horns. So in this case here, we break it up into different regions. So you got your posterior horn, your anterior horn, and your lateral horn. This is just showing you that particular location of, yeah, that's right. That's mostly a region that they designated as. I mean, it's like you could use the word component or anything like that. The horn's what they gave them, and I'm pretty sure they gave them because it does sound cooler. I think it was mostly based on the phenotype that they saw when they were first identifying these as well. White matter, basically you've got your three different horns here as well, your three regions. You got your posterior, your anterior, and your lateral as well, and they'll basically designate where you'll have most of that matter being associated. This particular, you should be able to look at something like this and really just have an idea about where these things are located. It's pretty easy to remember when you're talking about your horns. When we're talking about the functional organization of grey matter, though, it's really cool. Like, we actually can break up in terms of this is a cross section of spine, right? What you can actually see here when you look at the spinal cord is this particular region up here in the posterior part of the horn is going to innervate your somatic and visceral sensory nuclei, whereas your motor nuclei are actually broken up in terms of innervation between your visceral and your somatic sections for your motor function in the down between the lateral and anterior horn. So it actually is pretty cool that we know the actual specification of the spinal cord in regards to where impulses are going to go through and what's going to innervate what, which is pretty cool when you think about it. Dermatomes. I'm not going to expect you to remember every dermatome, but what I will expect is that you know what the definition of it is. Basically, it's regions of the body that are innervated by different neurons that span off different parts of the spinal cord.
Tell me,
There's some overlap going on with those as well.
So tell me.
But then again, like I said, you don't need to remember. If you don't go home and then start saying and start reminding yourself that for example, your right hip is innervated by L1 or anything like that on that dermatome. I'm not going to expect you to do that.
So it was about that.
You should be able to remember the reflex arc. The reflex arc is an autonomic response. And the way we do this is it involves if you remember these 5 things, you're going to be good for the exam, right? There's a very good chance, not a perfect chance, but a very good chance that you might actually be asked to describe the reflex arc and include all of the actual components of it. There you go, I've said too much. You should know that the impulse comes in through the sensory receptor, passes that information to the sensory neuron. Information gets processed to the central nervous system. That actually gets down, passes through the motor neuron to get the effector, which will be your arm if you've actually, touched something hot to pull off.
Go back to bed, then.
That's exactly what I just described. This is the level you should know this in as well. Like I said, I will expect you to know this. There is a 50-50 chance this is going to be on the exam in depth, right?
Alright, I'll talk to you
50-50, there you go, huh?
Love you.
But this is the particular step here as well. Here's the stimulus. The actual receptor picks it up, transmits it down down up through the spinal cord, goes up to the brain. Information gets processed by the brain, sends down some information down to motor neurons. This is going to go on and nerviate muscle fibers and then pull your arm back. Golly, how about that, huh? The reflex arc. Did anyone out know about the reflex arc before today? Or before this lecture? You did. You didn't? You did. You didn't. You did. Whew. Okay. Well, I hope those of you who already knew it liked learning about it again then as well. Who doesn't want to learn about that, right? Anyone who doesn't want to learn about that is it's a company I can do without. Okay, so the brain, a couple of things to remember here in regards to the brain. When you have to specifically talk about the efferent and efferent pathways when explaining it, you should know what they mean. Yeah, I mean, but the thing is, Georgia, as you go through this, right, you should be able to like define what they are, right? You know, in terms of what's coming in and what's going out, right? What's coming in are going to be your sensory receptors and your sensory neurons, and what's going out are going to be your effectors. Does that make sense? Yeah. Okay, regions of the brain, your cerebrum, your cerebellum, your diencephalon, and your brainstem. You've got to be able to locate these, and I'll show you where. First of all, your Geary, your sulci, and your fissures are basically, your fissures are the big grooves, sulci are the little grooves, and the Geary are the raised components of the brain. You should be able to locate that that's your cerebellum. This is your cerebrum, and the spinal cord coming down there at the bottom. You definitely need to be able to know that, right? I'm going to skip through here so that we can get into the good stuff. So I want to get into the brainstem over here. This is another part that you do need to know. You should be able to identify on the brainstem, the midbrain, pons, and the medulla oblongata. The midbrain is basically maintains your consciousness. Your pons basically is responsible for a lot of your motor control and your visceral function, and your medulla oblongata runs a lot of that sort of the autonomic stuff as well. That's just going to like blood pressure, heart rate, digestion, all this stuff that's going to be linked up and sort of work in maintain homeostasis by associating with endocrine functions as well. Things like, contraction of your smooth muscle tissue and stuff like that. Okay. Like I said, here's your diencephalon, your midbrain, your pons, and the medulla oblongata. This is a really good one for you to be able to remember, and a good one that if I put an image like this up in a quiz or in an exam, you should be able to identify the different regions. Okay. Like I said, your brain actually has meninges as well, has cranial meninges. It still has the same ones, your dura, arachnoid, and pia. These are continuous, as I mentioned before, with the spinal meninges. Okay, so I'll skip past this. We've got 19 minutes to get through the rest. I've already covered all that. And now we're on to the third component, senses, right? Pretty cool stuff. Come to your senses, everyone. Oh, I'm loving all the puns today. Absolutely loving the puns. Like just one lecture where I get to get really punned out. Does anyone else enjoy the puns? Am I the only one? Yeah. Fantastic. So every year, at the end of every year of my thanks guys. So every year, my feedback that I get at the end of the semester is usually that so I get people like James who tolerate them. They don't accommodate them though. I'd like to see that you accommodate my bad jokes, James, not tolerate them. But anyway, you can't help it, right? There every year the feedback that I get is that's good. I'm really glad to hear it. I either get things of Greg sucks or Greg's lectures are awesome. So I never get anything in the middle. I never get lectures were pretty all right. I never get pretty all right. I either get great or the worst ever. So, but that's okay. I can live with that. All right, let's get into it. This is you only got 3 learning outcomes for this one. I know you're all in the background. Like literally, some of you are doing the chainsaw at the moment. You're just punching down in the air. Like a cricketer who's just got a hat trick. Specify the components of the afferent and efferent divisions of the nervous system. Pretty easy with the stuff that is. Explain the receptors respond to stimuli and how the organization of the receptor affects its sensitivity. And then you should be able to identify the receptors for general sense and describe how they function. Bit of memory work coming up. Strap yourselves in, everyone. It's going to be one hell of a toboggan ride. Okay, you need to know this, right? And this all makes sense, right? If you remember one, then you kind of remember, the way I remember this when I was learning it is I remembered what my special senses were. I knew that, the main senses that we have. sight, smell, taste, balance, hearing, all that stuff was special. And then everything else that is just benign running in the background, temperature, pain, touch, pressure, vibration, proprioception, that sort of stuff is all general. That's going to keep running, right? Whereas this stuff is like you walk into the kitchen and you smell something good that's cooking and you go, oh, yeah. You don't really do that with pressure or anything or vibration. All right. So a couple of things to focus on for this section is, like I said, sensory pathways and your general senses, motor pathways, your somatic nervous system, and how it controls the contraction of the skeletal muscle. That's the main function of what we're talking about in this area. So sensory pathways, like we talked about before, these are ones that are not your general sort of ones. They're not just running in the background to keep you alive. These are ones where we're going to be able to get acute sort of interactions where we, through smell or sight or taste or anything like that as well. Your afferent and efferent components of this in regards to your somatic and visceral sensory pathways, where we that makes kind of sense when you go through this, right? The afferent division of this is largely going to be because that's your output, right? So that's going to be associated with the motor portion. So this is a really good example in regards to the way senses work. Here we have your basically let's put it all together. So here we've basically got the arrival of the stimulus. You get depolarization of the sensory receptor. This is like something that we want, a big stimulus, right? So it's an action potential generation. Immediately we get a big shoot up of information, propagates the actual axon, goes to the central nervous system, and then depending on what the actual stimulus was, will dictate what it's going to be. Is it going to be an involuntary motor pathway, or is it going to be to the voluntary motor pathway? The involuntary meaning that it's something that is going to immediately function in order to save your life, whereas the motor pathway function is going to be something where you can basically change or enhance. As it mentions here, there's only about 1% of arriving sensations that are relayed to your primary somatosensory cortex. So that's in regards to perception of if something's voluntary or not. You don't need to know that. That's just an odd fact. Your sensory receptors obviously detect the stimuli. Now, there's a couple of different things here. Transduction is basically the conversion of the arriving stimulus into an actual potential by a sensory receptor. This is really cool here. So receptors and receptive fields. I did say a part of the recorded lecture about when I used to teach this same subject at a different university. We used to have these things where basically if you imagined like a compass, and what they were is it was basically like an A, but you could actually shorten it in, right? So you can make it really narrow or you can make it really wide, almost like a compass, right? With two pointy ends. And what we used to do is we used to get people to look away, and they would say when they think they're experiencing 2 points touching or 1 point touching. And what happens is the more you narrow in these points, eventually you're going to get to a point where the 2 points the 2 pricks that they're getting are actually only touching onto the one receptive field. And that means that you're only going to perceive one bit of one touch, even though you're getting touched by 2 different things. Has anyone done that in high school or anything like that? I'm pretty sure they do that in year 12 nowadays. They didn't back when I was young, but has anyone done that experiment before? Yeah, Ava's done it. Nowadays, yeah, it's pretty cool. It's a great party trick as well, because a lot of people who didn't do science when they were in high school, you can actually wow and impress them, because a lot of people don't actually know this, and I think it's really, really cool. This is one of the reasons, and you have different composition of receptors or different amounts of receptors and receptive fields that are going to be in different parts of your epidermis, right? So if you think about it, your fingertips have a lot of receptive fields, and they're smaller, and they're all so that you can differentiate touch between the left part of your index finger and the right part, right at the tip of it. Whereas if you go to your back or you go to the side of your legs, you don't need that level of sensitivity. So they're going to be spread out a lot more, and the receptive fields are going to be larger. Adaptation actually will have 2 different forms. There's peripheral adaptation and central adaptation. Tonic receptors, you got to know the difference between all your different receptors. Tonic ones are slow adapting. As we can see here, tonic and phasic receptors are a little bit different. Tonic receptors are always active. So that's because there's action potentials that are generated in these particular ones. And that reflects like that level of stimulation that you end up getting. Phasic receptors are the opposite. These are fast adaptive. And that's because they're just bursts of energy, right? They're not normally active. The action potential will come in, but only at a short time in regards to the actual stimulus. Nociceptors and pain receptors are basically associated, obviously, with a lot of the there's no adaptation there, but they just pick up the actual sense, right? Thermoreceptors and temperature receptors are very, very interesting. They're super active when temperature's changing, and that's why you can actually respond immediately to this, which is really good reason, because our internal body temperature, as we've already talked about, needs to be maintained. One interesting fact, I'm wondering how many people knew this, but we have around 3 to 4 times more cold receptors than we do warm receptors. Did everyone know that? This is a cool thing. Like, you can be the coolest person at the dinner party, right? Because someone will say, "Oh, I always get cold." This is how I met my partner. I just said to her, she said, "Oh, I always get cold." And I'm like, "That's because you have 3 to 4 more cold receptors than you do warm receptors." And then she said, "Wow, you know a lot, don't you?" And I said, "Oh, not at all." And then and that was it. We went out on a date. So, but seriously, there is actually, that's the reason why so many people say they don't like the cold. You don't get a lot of people saying they don't like the hot, right? That's because we pick up on that change in cold temperature much more than warm temperature, right? Hypothermia is a thing, right? And we need our body to be able to respond to it and adapt quite quickly. Chemoreceptors. So these will detect small changes in concentrations of specific chemicals, mostly pH and gas exchange with carbon dioxide and oxygen. Okay, mechanoreceptors, as it mentioned, these are for mechanical movement, stretching, compression, twisting, anything else that's going to stuff around the membrane, right? Okay, we'll go into these a little bit right now. This will be the last part of the lecture. So tactile receptors, these are associated with your touch, pressure, and vibration. Baroreceptors and proprioceptors are going to be associated with your internal visceral sort of function with baroreceptors and proprioceptors. Easiest way that I remembered this when I was learning about it was baroreceptors. I always immediately just thought blood pressure. Because, internal barometer, and then everything else that came along with visceral function, and proprioceptors, that just refers to the way that your body's proportioned, right? So it makes sense in regards to thinking that that's going to be around your joints and skeletal muscles. But you should be able to have, just remember the basics of these. I'm not going to put quick pictures of these up in the exam and say, is this a free nerve ending or not? But you should know the basics of how the 6 type of tactile receptors. So root hair plexus, tactile discs, your bulbous corpuscles, the laminar corpuscles, and the tactile corpuscles. You should just remember what they're for. You don't need to remember every one of these dot points. I just want you to remember what they're actually for, right? In this case here, tactile corpuscles, fine touch and pressure, laminar corpuscles, deep pressure, bulbous corpuscles, super sensitive to distortion of the skin, tactile discs, fine touch, and root hair plexus, well, that's a no-brainer. And free nerve endings is super sensitive to touch and pressure, right? That's all I want you to remember, but I want you to remember all things, okay? We got a deal? All the touch and pressure, yeah, but they're all at different situations as well. Some are for deep pressure, some are for pressure that's going to be like on the surface of the skin. It really comes down to where these things are located, okay? So we got a deal, if everyone can remember that. Fantastico. All right, everyone. That's all we've got time for. Make sure you don't miss out on your lab 2s. Some of you have on your timetable that you've got 2 separate weeks, right, for lab 2. That's because of the Easter break, right? So what I want you to do is, some of you are broken up into 1 week, some into the other, right? Go and check with Student HQ. Like I said, I can't access your timetable. Yeah, so Nicole, I mentioned this early on, right? So we've released the ones for quiz 2 and quiz 3. There's some people still doing quiz 4. So while there's other people doing a quiz, I'm not going to release the marks just because that opens the door to cheating. Yeah, so that should be, I should be able to open that next week. Yeah, I've got a couple of past exams, Treon, and I'll put them up probably next week as well. I'm actually writing the lecture for next finishing off, putting the finishing touches on the revision lecture that we're going to do in a few like in around week 11. But you know, I'll make sure that the past exams are all accessible way before then anyway. Aiden, I'll find out when you find out. I find out a day or 2 before you guys do, so I don't know what part of the exam period it's in. Can you reattempt past quizzes? No, Esther. One go only. They're one show only. Yeah. You got to check the instructions before you start. If anyone, so in regards to when we missed labs, if we still do the worksheet, you lose percentage. You can't submit labs that you haven't attended, Akira, unless you had special consideration. So that's because it's just the part of the units accredited. So we can't have people doing actual on class. You did a special. Send me an email then, Akira. I know you have, but yeah, like I said, you get bogged down with so many emails. For the reading, should I still read it if I understand everything from the lectures? Not unless you probably don't need to. The lectures, the readings are to enhance the lectures. So the lectures are basically a little synopsis of the actual textbook itself. So if you're feeling really comfortable with this is what I'm basing the exam off. So there's not going to be extra information unless I've mentioned in a lecture and said, this learning objective, you're going to have to go to the book for that because we don't cover it in this because we don't have space. Then that's the only ones that you should go through. All right, everyone, that's all we got time for. Speaking of quizzes, is there any way you can put more questions up on Twitch? I don't do Twitch, Aiden. So Twitch is a new thing from the university. Never existed, and that's run by the Twitch people. They basically look at my content and they come up with revision stuff for you guys. So yeah, that's, I can give feedback though, for sure. But yeah, I've as far as I'm concerned, you guys have enough content here already. Not a huge fan of Twitch, but if it helps, then give it a go. All right, everyone, that's all we've got time for. Have a great day, and I'll see you all in the next one. You're very welcome. See you, everyone.